Hello students, in this practical lecture we will learn about cell suspension culture. Let us start with a brief introduction. A cell suspension culture is the culture of single cells, small cell groups or larger cell aggregates in a liquid medium under agitation and aeration. Cell suspension cultures are generally initiated by culturing small masses of callous tissues in liquid medium, which is then agitated during the culture period. The growth curve of a typical plant cell suspension culture consists of four essential stages: an initial lag phase, an exponential or logarithmic phase, linear phase and stationary or dead phase. The duration of each phase of growth is dependent on the species or genotype selected, explant use, culture medium, size of inoculum and the time of subculture. Growth in suspension cultures can be monitored by determining the cell number, packed cell volume, cell phase and dry weight or optical density or OD. Suspension cultures can be used to study significant events about cell physiology, biochemistry and metabolism at the cellular level. Suspension cultures can be used for production of useful secondary metabolites also. Cell suspension cultures can also be used to produce mutant cell clones by mutagenesis, from which mutant plants can be regenerated. Cell suspension cultures are commonly established from sterile seedling explants or imbibed embryos or young leaves. Explants derived from seedling, example hypocotyl, cotyledon, etc. and leaves can be used to start cell suspension cultures by mechanical method. The soft tissues are generally grounded in a hand-operated glass homogenizer and the homogenate of intact living cells, dead cells and cell debris is filtered and centrifuged. The homogenate is then transferred to liquid medium and maintained in a shaker. On the other hand, cell suspension cultures are commonly established from callus cultures. For this, callus is first induced from the young explant tissues and friable calli obtained are then cultured in liquid medium to obtain suspension cultures. The time of subculture and dilution required for subculturing are determined by studying the growth pattern of the culture, and cell suspension cultures are maintained by subculturing at regular intervals. The major equipments required for this experimentation include shaker, centrifuge, autoclave, pH meter, weighing balance and laminar airflow. Next, let us see the reagents and other requirements. Reagent: glass bottles, spatula, tissue rolls, distilled water, cotton plugs, aluminum foils, scalpel handles, sterile blades, forceps, borosil glass test tubes of the size 150 mm into 25 mm without rim preferably, and conical flask of 250 ml. Containing Murasige and Skooks or MS 1962 medium, marker pens and green pea seeds. Let us see the procedure of the experiment. First, rinse the green pea seeds under running tap water and then put the plant material in a jar with screw-capped lid containing water and wash with 0.1% detergent for 3 to 4 minutes. Gently agitate the content of the jar every 20 to 30 seconds during the washing step. Next, pour off the detergent and rinse the plant material with tap water and cap the container. Next, spray the container with 70% ethanol and bring it to the sterile workbench of the laminar flow. Now spray the sterile workbench of the laminar flow with 70% ethanol and also spray vessels and all tool packets containing petri dishes, forceps, scalpel handle, razor blades etc. with 70% ethanol before placing them on the work surface. Now remove the cap of the container containing the plant materials and pour 0.1% mercury chloride solution and replace the cap. Wash the plant materials by intermittent sacking for 6 minutes. Now pour off the mercury chloride solution and pour sterile water over the plant materials and rinse the plant materials for 2 to 3 minutes by gently agitating. Repeat the rinse with sterile water for 3 to 4 times. Using the sterile forceps, transfer the seeds in a sterile petri dish and peel off the seed coat using a sterile blade. Use the separated cotyledons as explants. Now open the cotton plug of the culture tube containing MS medium supplemented with 3 mg per liter 2,4-D and flame the mouth of the tube. Flame the forceps and cool them by touching the medium in the culture tube. Use the forceps to hold explants and place it on the medium by gently pressing so that the explants make contact with the medium. Replace the cotton plug of the culture tube and mark the tube with date and details of the experiment. Now incubate the culture tubes in a growth chamber provided with 16 hours light and temperature maintained at 25 plus minus 1 degree Celsius for 5 to 6 weeks. Observe the cultures every week. After about 2 weeks, calli may start appearing on the surface of the explants and friable calli will be formed after about 4 weeks. The growth curve for the callus can be established by measuring fresh weight in 4 days interval during a 32-day period followed by subculturing every 32nd day. Cell suspension cultures were initiated from the callus on liquid medium containing 3 mg per liter 2,4-dichlorophenolic acid or 2,4-D. Cell suspension cultures were established by inoculating 0.5 gram of 4-week-old friable calli in 25 ml liquid MS medium supplemented with 3 mg per liter 2,4-D. Cultures are agitated on the rotary shaker at 125 rpm and maintained in complete darkness at temperature of 25 plus minus 2 degrees Celsius. The biomass growth of the cell suspension cultures were studied for a growth cycle comprising of 32 days by subculturing about 2 grams of cells on 50 ml of fresh medium after every 32 days. Growth curve of cell cultures was studied by sampling at an interval of 4 days during the 32 days period of growth cycle. The growth of cell suspension culture was measured in terms of packed cell volume, gram fresh weight and gram dry weight. To determine the fresh weight, cell suspension was filtered through filter paper and biomass was weighed. To determine the dry weight, the pre-weighed cell was dried in hot air oven at 50 degrees Celsius to a constant weight and recorded. To determine the packed cell volume, the cell suspension was transferred to a graduated 50 ml flat bottom centrifuge tube and was allowed to settle for 1 hour. Then the volume occupied by cell was recorded and result was expressed as packed cell volume. Next, let us see the results. Calli started appearing on the surface of explants after about 2 weeks of culture, and friable calli were formed after 4 weeks of culture. Further multiplication of callus tissue was achieved by subculturing the calli on the same media after every 32 days. Suspension cultures with cell clumps were observed in the liquid medium after 4 weeks of culture. The growth curve of the cell suspension cultures consisted of an initial slow lag phase comprising of the first 4 days, followed by an exponential growth phase ending at day 15. The cultures then entered a linear phase of growth from day 16 to day 23, followed by a final stationary phase from day 24 to day 32. The maximum increase in cell biomass was obtained on day 24 of growth cycle, reaching up to 7 gram fresh weight per culture. Thank you.